afternoon. I'd like to thank the Human Frontiers Science Program organization for the invitation to Japan. It's my first time here, and it's an amazing and lovely country and amazingly nice people. I'm going to tell you today about our journey studying the immune system of bacteria. I know that for some of you it may sound weird that bacteria have an immune system, because immune systems are usually used by us to protect against bacteria, but in fact bacteria need to protect themselves very frequently against viruses that infect them. Viruses that infect bacteria are called phages, and these are the most abundant viruses on Earth. Although they don't infect us, they infect the most abundant organism on Earth, which is bacteria. And usually they will just find their bacterium, inject their DNA inside the bacteria, and replicate in the bacteria, eventually killing it. And altogether, if you think about these viruses are so small, you can only see them through electron microscope. But if you weigh all of them, if you take all these phages and weigh them, they weigh 4 times as much of all humanity together. So they're pretty important in the balance of Earth. So to protect against infections, bacteria evolved a very sophisticated and fascinating immune system. And if you looked at the immune system of bacteria until maybe a decade ago, you'd think mostly about 2 main arms of immunity. One arm of the innate immune system of bacteria is mainly comprised, or was thought to mainly comprise, of restriction modification systems, which are enzymes, restriction enzymes that cleave the DNA of the phage in specific sequence motifs, and at the same time protect the DNA of the bacterium against cleavage. And the more recently discovered system, the adaptive immunity arm of bacteria, is called CRISPR-Cas. And it's a fascinating system that allows bacteria to memorize past infections of phages by storing pieces of DNA from the phages inside the genome of the bacteria, and then using these pieces to identify future infection by the phage and kill them. But as I told you, about a decade ago, we started thinking that this might not be the end of the story, that there might be some new defense mechanisms that are not known yet. And the reason we hypothesized this is because the CRISPR-Cas system was discovered really late in the history of biology. CRISPR-Cas was discovered 90 years after the discovery of the phage, and throughout these 90 years, phages and bacteria have been studied very extensively. And the reason why CRISPR was not discovered until late is because it's not very active in E. coli, and that's the main organism where people studied phages and bacteria. So we thought that there might be other defense mechanisms in non-model organisms that could protect against phages. Then we thought of how we would look for them. We had multiple ideas of how to look for them. Almost none of them worked, so a lot of failures before things started succeeding. But what eventually worked for us really well comes from a very peculiar behavior of defense systems when you look at genomes of bacteria. Because in genomes of bacteria, these defense systems aggregate together. If you look at genomes, the genes that code to restriction enzymes are very frequently coded next to CRISPR-Cas systems, and that you see that again and again in many different genomes, and these are the yellow boxes that you see here code for genes encoding defense systems. So we started thinking that if we will look for cassettes of genes of unknown function that are very frequently found next to CRISPR and restriction enzymes in many different genomes, we might find new defense systems. We looked for defense genes or candidate defense genes in defense islands. And we looked for them in tens of thousands of microbial genomes. And one of the key problems that we bumped into at the time was that we just found too many of these genes. There were about 2,000 candidate defense systems or candidate cassettes of genes that we found next to known defense systems. And at that point, we were in a little bit of a loss, because it's very difficult to study 2,000 different kinds of gene cassettes. So we did something quite extraordinary in science. Instead of, as you know, all of you who've done your PhD, you know that the ethos of science is that there is one PhD student or postdoc that runs with the flag and conquers, despite the difficulty, conquers the peak of the mountain and gets the praises. But this ethos really prevents real teamwork in science. And here we try to overcome that. So we pooled the brains of all the people in the lab. This you see the people in the lab at the time, 7 years ago. We called ourselves the Defense Council, looking for defense systems of bacteria. And each lab member received a few hundreds of candidate defense systems to consider and prioritize for further research. And these were long days, but very fun. I fed them with pizzas. It always works with PhD students. If you want my advice, use pizza for science. And here you see that the light in the room is a little bit different, so it's the afternoon. And eventually, everybody who participated in this endeavor really remembers it very fondly, because a lot of fantastic discoveries came out of this room. So here, these are the people that eventually published papers that became key papers in the field. So at the same time, where we predicted defense systems, we had to develop an experimental way to test them experimentally. And we do that quite simply. We put them in bacteria that don't have much defense systems, bacteria in the lab, E. coli and Bacillus subtilis, and then we infect them with a series of phages to see whether the bacteria are now protected. And when we developed this process, it took us about 1.5 years or 2 to develop the concept, both the computational and the experimental part. And we were thinking that if we will discover maybe 2 or 3 new defense systems, this will be marvelous. But in fact, we were extremely surprised, and I'm still surprised to date, that this endeavor and the endeavors that came after that from my lab and from other labs that were inspired by these kind of screens revealed that there are more than 250 defense systems that were completely unknown to us at the time. And so this is a pretty complicated picture. At the time when we discovered them, these were just genes or boxes that describe genes of completely unknown function. We started calling them by acronyms. So the first system we discovered was called BREX. That stands for bacteriophage exclusion. And the second system was DISARM, which is Defense Island System associated with restriction and modification. But then we ran out of acronyms, so we decided to call these defense systems by names of protective deities, protective gods from different mythologies. And for the local scientists here, you probably recognize the name of Hachiman, one of my favorite defense systems, which was eventually solved by the lab of Jennifer Doudna a year ago. So at the time, we were very heavily invested in genomics and metagenomics, but clearly it became clear to us that if we want to really solve mechanisms of action, we need to transform ourselves into becoming much more biochemically heavy lab. And I did that through hiring students and postdocs who were really strong in that aspect. And we started deciphering the mechanisms of defense and really revealed a wonderful zoo of functions, defensive functions that bacteria have against phages. So we discovered defense systems that use cyclic dinucleotides, small molecules, to signal internally in the cell when infection is sensed. We discovered defense genes that produce toxic small molecules, so small molecules that are poisonous for the cell cycle of for the replication cycle of the virus and inhibit the replication. These molecules are now, we're testing them whether they can be used as antiviral molecules for humans, for viruses that infect us. We discovered a defense system that relies on non-coding RNAs that become partially reverse transcribed and form a bait for the phage. And when the phage inserts itself into the cell and bites into this bait, it goes off like a bomb and releases a toxic effect. Really fascinating. We discovered giant complexes that are formed in the cell, giant defense machines. This one is 4 times larger than the ribosome, and it forms in the cell to protect it during phage infection. Discovered new signaling molecules. We discovered defense systems that were the ancestral defense systems to a very common and central plant immune mechanism. We discovered a defense system that is ancestral to the human inflammatory pathway called pyroptosis. Discovered systems that use ubiquitin-like proteins to ligate ubiquitin-like proteins on the virion to interfere with the viral assembly. Really a zoo of wonderful machines. One of the things that really surprised us is that many of the machines we discovered turn out to be ancestral to key components, to key pathways that we already know are present in human cells, in animal cells, to protect against viruses. I'll give you one example. This is the SIGA-STING pathway. It's a pathway that is present in human cells, in animal cells, and allows these cells to recognize foreign nucleic acids. And when nucleic acids are recognized, this pathway generates a specific signaling molecule comprised of two nucleotides bound together, A and G together. And this signaling molecule activates a second protein called STING, and this STING activates the downstream immune response. We discovered that bacteria contain the ancestral form of the SIGA-STING pathway, a protein that is a homologue, a distant homologue of the human SIGAs that senses foreign nucleic acid that come in with the phage, generates the signaling molecule that activates the STING form of the bacterial defense system that generates immunity. And this is just one example. We found that multiple components, I think by now we know of 10 or 12 different components of the innate immune system of humans that evolved from the bacterial immune system. This is just a partial list for the immunologist within RNAi, inflammasomes, pyroptosis, node-like receptors. All of these we find in bacteria, and we realized that these elements evolved in bacteria way before there was even a first eukaryote. The first eukaryote evolved after these bacteria had defense mechanisms to protect against phages, and then this first eukaryote, which evolved from a fusion between an archaeon and a bacterium, inherited these defense mechanisms from the bacteria and the archaea. And this understanding now allows us, finally, to start understanding eukaryotic immunity by using bacteria as model organisms for studying immunity, which is something that, if you told this to me a few years ago, it would have been incredible. So just one very simple example, we discovered a defense mechanism, a protein domain, so a piece of a protein that bacteria use to protect against phages, and when this domain comes into action, when infection is recognized, this protein domain cleaves the molecule ATP and eliminates ATP from the cell. ATP is the very central molecule for the energy economy of the cell, and without ATP, nothing can replicate. Of course, the viruses cannot replicate as well. After we found them in bacteria, we discovered the same protein domains in immune proteins of in animal cells, and we showed that these domains also degrade ATP. And that's not the only case, so we discovered other defense mechanisms. This is too complicated to explain in a short presentation, but we discovered a mechanism of defense in bacteria that relies on protein domains and small molecules that were later discovered to be super important or very central in the immune system of plants, how plants protect themselves against plant pathogens. So by now we know that there are more than 250 different defense systems that bacteria and archaea use to defend against phages. This is a very complex picture where we thought to put all of what we knew back 3 years ago on one poster. Of course, this is completely obsolete right now because since then our lab and other labs around the world discovered many more defense mechanisms. But some of the very rewarding aspects of this discovery is that although none of these systems existed in the model E. coli organism in the lab, we now know that nearly every bacterium whose genome was sequenced have at least one of these defense systems, and often more than once in its genome, making this a universal feature of bacterial biology, which is, again, to the young students of you, or for the budding researchers in the audience, I know how it is when you start your research, you feel that everything has been discovered, and you're entering the world where there's nothing substantial to discover yet. And here's an example that we really don't know much. So especially if you go to study non-model organisms, there are very good chances that you'll bump into something unknown. So all this list of the things in the right are names of defense systems whose mechanism was not deciphered yet. So a lot of work to still be done. One of the surprising parts, and I'm nearing the end here, is that bacteria could be used as model organisms for studying animal and plant immunity, and we and others are doing that extensively these days, and studies not from my lab, also from other labs show amazing discoveries based on bacterial immunity, which teaches us on human and plant immunity. And the last part of this presentation is just to show how science evolves, and again, a very rewarding aspect of the research is what was our own gem, so the thing that we were the only ones probably interested in back 7 years ago, this room full of interested people eventually grew to be a world full of interested people. So this is from a conference we've done on the immune system of bacteria in Paris this year, and now there are thousands of people that are interested in these same defense mechanisms. So arigato gozaimashite for your listening. Thank you very much.